Bismillahirrahmanirrahim. This is Dr. Askam. Hope that you are doing well. So today we move to a new stage of embryonic development, which is segmentation stage. This stage comes directly after the fertilization. So let's remind that after the fertilization, we at the end of the fertilization we have formed a zygote. Okay, so the segmentation is the transformation of the zygote in a multicellular from the unicellular state to multicellular state. Okay, this happened through a series of mitotic divisions in order to form a multicellular structure called blastula. Okay, so here the zygote will divide using mitosis, and at the end of a series of these divisions, it forms this formation, which is blastula. Okay, so let's follow this example. It is an example of an human egg. So after the formation of the diploid nucleus of the zygote will start to divide, firstly in 2 cells, after that in 4 cells. At the 3rd day of its development, it transforms to a mass of cells called morula, and then after this morula will transform to blastocyst, which is a multicellular structure composed from a series of cells which surrounds void. This void is called blastocell. Okay, so let's follow this series of divisions. But before this, you have to know that there are many types of segmentation. In fact, these types depend of the size of the zygote which divides. So the zygote or the egg, in the nature there are many types of egg according to their composition of yolk, of special compound called yolk. This yolk, or you can call it also yolk reserves, are in fact proteins, carbohydrates, and lipids. So why the zygote will take yolk in its cytoplasm? It's in order to develop its embryo. So if the development is inside the body, the zygote don't take yolk, but if the development is outside the body, so naturally the zygote will take yolk. But in nature there are many types of eggs according to their the quantity of the yolk in their cytoplasm. So let's start by the first one, the allicate egg, which don't contain yolk. Okay, it's the case of mammal's egg, because the development of mammal's embryo are inside the body. Okay, the second one is oligolycite egg. Here we find a few quantity of yolk. You have several examples, such as sea urchin egg. Number 3 is heterolycite egg. So in this case, the yolk is the quantity of the yolk is very important, and it is concentrated in the cytoplasm. Okay, so here you have several examples, such as amphibian egg and fish egg. Okay, so number 4, centrolycite egg. Here in this case, the quantity of the yolk is important, such as heterolycite egg, but it is around the nucleus. So the yolk is concentrated around the nucleus. That's why we call it centrolycite egg. Okay, let's take an example such as all insect eggs, like fly. So here in this case we find an important quantity of the yolk, but it is concentrated around the nucleus. After that, number 5, which is the egg that contain the most important quantity of the yolk. So here it's called telolysis egg, and it contains a very important quantity of yolk, like the case of bird eggs or chicken egg that we consume in our every day. Okay, so according to this quantity, we have two types of segmentation. Okay, so if the egg is allicite, oligolycid or heterolycid, the segmentation is total segmentation, or what we call holoblastic segmentation. Okay, and this in this case, so the totality of the egg will divide into 4, 8, etc. So all the divisions are totally all the volume of the egg divide in 2 or in 4 or in 8, etc. So that's why we call it total segmentation or holoblastic segmentation. Okay, this segmentation can be radial or spiral or rotational, and I will show you the 3 kinds of total segmentation after. The second type of segmentation is partial segmentation or meroblastic segmentation. So it is the case of the centrolysid egg and telolysid egg, which are charged in yolk. So it can they can divide totally. It's only a part of the cell that make all the steps of segmentation. So that's why we call it partial segmentation or meroblastic segmentation, and this segmentation can be discoidal or superficial. So let's see the first example, which is total segmentation or holoblastic segmentation. So as I said before, in this case the totality of the cell will divide. Okay, in order to assure to all the steps of the segmentation from the step 2 to morula and to blastula. So this segmentation can be radial. So in this case the cleavage plane it's like this, like you can see here. It's the case when it is equal. So at the step 2 we obtain 2 cells, 4 cells, 8 cells, etc. until morula, and you can see that the cleavage plane are in the same state. Okay, here it is unequal. So at the stage 8 you can see that we obtain small cells and big cells, but anyway it is total, and the cleavage plane are in the same direction. But here in the case of spiral we obtain especially we can see it at the stage 8. So you can see that the blastomeres are decaled. Why? Because the cleavage plane is not in the same direction. Okay. And finally rotational. So in this case the blastomeres will divide in 3 dimensions. Okay, so that's why we say rotation. After that, from the partial segmentation or meroblastic segmentation, so in this case it can be discoidal, like a disc. Okay, it's the case of bird egg. Okay, so as a disc, we call it discoidal, and at the final step we obtain germinative disc. Okay, but in the case of insect egg, it is superficial, so it's only, as you can see, it's only a part of the cell that divide. Here it is this part, and here it is only the nucleus that divide. Okay, because it is charged in York. So I hope that you understand me. Let's take now let's take two examples. We will take the first example concerning the total segmentation, and then we will talk about the second example, which is partial segmentation. So for the total segmentation, we will take oligolycid egg as example, and we will follow its series of divisions from the stage 2 to the stage 16. As example, we will take C urchin. So the C urchin, as you can see, it is composed from several things. Okay, it is oriented firstly, so animal pole and vegetative pole. I didn't translate this picture. And then how we can recognize this orientation. It's according to the nucleus, which is close to animal pole, and for the urchin, for the sea urchin egg, before fertilization, it contained the orange pigmentation in all the surface, on all its surface, but after the fecundation or after fertilization, this pigmentation moves in the vegetative pole. So that's how we recognized fertilized egg. Okay, so let's say that we have fertilized the egg, and we will try to follow it from the first division to the end of the segmentation. Okay, so let's discover the cleavage plane. So here, when the zygote, the first division of the zygote is in two cells. Okay, and this division is according a specific plane, which is meridian plane. So as you can see, the meridian plane is a plane that moves from animal pole to vegetative pole. Okay, and it's divide the zygote in two cells. Okay, so the result is here. We obtain two identical cells, and each cell is called blastomere. Okay, so it is the stage 2 because we have two blastomeres, and these blastomeres have the same size. Okay, after that, the two cells, the two blastomeres, will divide again in two in order to obtain 4 blastomeres. So the cleavage plane is also meridian, but here it is perpendicular to the first plane. So if I take the first plane like this, I must cross it perpendicularly in order to divide my two blastomeres into 4 blastomeres. Okay, so here we obtain 4 blastomeres, 1, 2, 3, and the fourth is in the other side. Okay, so this is the second division. After that, the third division will divide my fourth blastomeres in two. Okay, so the plane, the cleavage plane here change. Okay, it is equatorial and not meridian. equatorial, it means that it crosses the cells in the middle, okay, like this. And at the final we obtain 8 blastomeres. When I say blastomeres, it means that all the cells have the same size, and it is the stage 8. Okay, so after 8, what's happened? After 8, the segmentation will change between the cleavage plane will change between the animal pole and the vegetative pole. So let's start by the animal pole. In the animal pole we have 8 blastomeres, so each blastomere will divide into according to meridian plane. Okay, so each blastomere will divide into according to meridian plane. I mean, each blastomere will divide in 2. Okay, in order to obtain 8 equal blastomeres, and each one is called mesomere. So we obtain 8 mesomeres. Okay, so that is what's happened in the animal pole. But in the vegetative pole, it's the cleavage plane. It's is different. Okay, so here the 4th blastomeres will divide in according to one cleavage plane, which is latitudinal, eccentric toward the vegetative pole. So it is latitudinal, okay, so in large and not in long, and also it is eccentric toward the vegetative plane. Okay, so here in the vegetative pole we obtain 4 big cells and 4 small cells. So the big ones are called macromeres, and the small ones are called micromeres. Okay, so we obtain 8, but the 8 are not equal. There are 4 macromeres, which mean 4 big cells, and 4 micromeres, which mean 4 small cells. Okay, but when we but at the end we obtain 16 cells. Okay, so 8 mesomeres, 4 macromeres, and 4 micromeres. Okay, after that, after the stage 16, the 16 cells will divide into to form 32 cells. Okay, and the 32 cells continue their divisions, and we obtain a mass of cells, and this is what we call morula. Okay, and then the morula will transform to blastula, exactly to a regular celloblastula. Okay, and this celloblastula is composed from a series of cells which surrounds a void, which is blastocell. Okay, so this is our first example, which is the total segmentation of oligolycid egg of the sea urchin. Now let's move to the second example, which is partial segmentation, and we will take an example of telolycid egg, exactly the chicken egg. Okay, so we'll follow how it is transformed from a cell, which is in this part, to this formation. Okay, so let's start by the stages. So here we have a polar view of our egg. Okay, so here you have the yolk, and here you have the part which divide, and which is called cellular part. Okay, so cellular part will start its divisions. Okay, so the first division will divide this space in two, and then the two will divide in four, the four will divide in eight. After that, it continues until the formation of this formation, which is called blastodisc. Okay, so let's follow. So here the stage 2, the stage 4, the stage 8, the stage 16, and then at the end of the segmentation we obtain a blastodisc. So the blastodisc, when we observe it in microscopy, it look like this. Okay, it look like this, and it marks the end of the segmentation. Okay, so we have finished with our two examples. I hope that you understood me. Let's stay in contact by Telegram and Facebook as usual, and I am waiting for your commentaries, and see you in the next video for the next stage of embryonic development. Thank you for your attention.